Dear learners, I am Ramya, working as Assistant Professor in the Department of Biotechnology, St. Joseph's College of Arts and Science for Women, Hosur. We are going to learn about culture techniques. What is culture technique? A microbiological culture or microbial culture is a method of multiplying microbial organisms by letting them reproduce in predetermined culture medium under controlled laboratory condition, that is in vitro condition. Microbial cultures are foundational and basic diagnostic methods used as a research tool in molecular biology. Microbial cultures are used to determine the type of organism, its abundance in the sample being tested. The term culture is more generally used informally to refer selectively growing a specific kind of microorganisms in the lab. To isolate a pure culture of microorganism, a pure or axonic culture is a population of cells or multicellular organism growing in the absence of other species or types. A pure culture originates from single cell or single organism, in which case the cells are genetic clones of one another. For the purpose of gelling the microbial culture, the medium of agarose gel agar is used. Agar is a gelatinous substance derived from seaweed. A cheap substitute for agar is quargum, used for the isolation and maintenance of thermophiles. Bacterial culture. There are several types of bacterial culture methods. First one is broth culture. One method of bacterial culture is liquid culture, in which the desired bacteria are suspended in a liquid nutrient medium in an upright flask. This allows a scientist to grow up a large amount of bacteria. Second one is placing the bacterial culture in agar plates. Microbiological cultures can be grown in petri dishes of different sizes that has a thin layer of agar-based growth medium. It can be incubated at an optimal temperature for the growth of selected bacteria. Agar plates can be stored upside down in a refrigerator for an extended period of time to keep bacteria for future experiments. Next method is agar-based dipsticks. Miniaturized version of agar plates implemented to dipstick format. Example, we can say dip slide. The other method is stab culture. Stab culture are similar to agar plates but are formed by solid agar in a test tube. Bacteria is introduced by an inoculation needle or a pipette tip being stabbed into the center of the agar. Bacteria grow in a punctured area. Stab cultures are most commonly used for short-term storage or shipment of cultures. Solid phase culture of thermophilic microorganism. For solid plate cultures of thermophilic microorganisms such as Bacillus acidocalcareus, Bacillus stereothermophilus, Thermus aquaticus and Thermus thermophilus, etc., growing at temperature of 50 to 70 degrees Celsius. Virus or phage culture. That can be a required host cells in which the virus or phage multiplies. For bacteriophages, cultures are grown by infecting bacterial cells. The phage can then be isolated from a resulting plates in a plate. Virus cultures are obtained from their appropriate eukaryotic host cells. Why this culture technique is important? Pure cultures are important in microbiology for the following reason. One, purified the isolated cultures can be cultivating, understanding only the desired microorganisms in growing in the media. Pure culture can be identified accurately after testing the organism. Spontaneous mutation rate will be low and same results can be ensured and it will be 99.9% identical. Who discovered this pure culture technique? Louis Pasteur. He is called as father of microbiology. Robert Koch was one in this pure culture technique called as father of practical bacteriology. And agar was discovered in the year 1658. Agar was first used in microbiology lab in the year 1882. So how this isolation of pure culture done? Culture composed of cells arising from same progenitor cell. Progenitor is termed as CFU, that is colony forming unit. Aseptic techniques prevents contamination of sterile substance or objects. So what four types of aseptic techniques: streak plate method, spread plate method, pour plate, and serial dilution method. Streak plate method. This method is used most commonly to isolate pure cultures of bacteria. A small amount of mixed culture is placed on the tip of inoculation loop or needle and is streaked across the surface of agar medium. The successive streaks thin out the inoculum sufficiently and the microorganisms are separated from each other. These plates are incubated to allow the growth of colonies. Because of this dilution gradient, confluent growth does not take place on that part of the medium where few bacterial cells are deposited. Progeny of single microbial cells represent a clone of pure culture. This is the method of streaking. There are various methods of streaking. One is T-streaking, continuous streaking, etc. Quadrant streaking. Next is pour plate method. This method involves plating of diluted samples mixed with melted agar medium. The main principle is to dilute the inoculum in successive tubes containing liquefied agar medium so as to permit a thorough distribution of bacterial cells within the medium. The mixed culture of bacteria is diluted directly in tubes containing melted agar medium maintained in the liquid state at a temperature of 42 to 45 degrees Celsius. The bacteria and melted medium are mixed well. This is a diagram of pour plate method. Next is about spread plate method. The mixed culture or microorganism is not diluted in the melted agar medium. It is rather diluted in a series of tubes containing sterile liquid, that is water or saline. A drop of so diluted liquid from each tube is placed on the center of the agar plate and spread evenly over the surface by means of sterilized blunt glass rod. The medium is now incubated. When the colonies develop on the agar medium plates, it is found that there are some plates in which well-isolated colonies will grow. This happens as a result of separation of individual microorganisms by spreading over the drop of diluted liquid on the medium of a plate. The isolated colonies are picked up and transferred onto a fresh medium to ensure purity. This is the diagram. This is a diagram for spread plate method. The mixed culture can be formed as isolated colonies. Next is serial dilution method. This method is commonly used to obtain pure cultures of those microorganisms that have not yet been successfully cultivated on a solid media and grow only in liquid media. The aim of this dilution is to inoculate a series of tubes with a microbial suspension so diluted that there are some tubes showing growth of only one individual microbe. If we take out 1 ml of the medium and mix it with 9 ml of fresh saline liquid medium, we would then have 100 microorganisms in 10 ml or 10 microorganisms per ml. If we add that 1 ml of the suspension to another 9 ml of fresh sterile liquid medium, each ml would now contain a single microorganism. That's how the serial dilution method takes place. This is the diagram of serial dilution technique. Next is about anaerobic culture methods. We can see these anaerobic culture methods in brief. An anaerobic organism or anaerobe is an organism that does not require oxygen for growth. It may react negatively or even die if oxygen is present. It may be unicellular, example of anaerobes, protozoans, and some kind of bacteria, or multicellular organisms. An anaerobic bacterial culture method used to grow anaerobes from a clinical specimen. Obligate anaerobes are destroyed when exposed to atmosphere for as briefly as 10 minutes. Some anaerobes are tolerant to small amount of oxygen. What are all the suitable specimen for anaerobic culture? Blood, CSF, that is cerebrospinal fluids, dead tissues, endometrial swabs, sterile surgical samples. Unsuitable specimens for anaerobic culture are normal flora samples like urine, mucus, sputum, saliva, and fecus. These are all the unsuitable specimens for anaerobic culture. Anaerobic specimen transport must be done immediately to the lab within an hour or 2 hours. Never refrigerate sample for anaerobic culture because if it exposed to the oxygen, the sample will not work out. There are main four methods of anaerobic culture method: Robertson cooked broth, culture away from oxygen, chemical exclusion of oxygen, mechanical exclusion of oxygen. Question for today's learning is: what are all the unsuitable specimen for anaerobic culture methods? Thank you, dear learners.